Good afternoon, let's start the lecture. Second unit. The circulation. Chapter 3. The normal electrocardiogram. Study of electrical properties of the myocardium. It is imperative that you familiarize yourself with this topic with the help of a textbook. Guyton and Hall. Textbook of Medical Physiology. Pages 101 to 115. This lecture can be found on the Normal Physiology course website. Overview of the circulation, medical physics of pressure, flow, and resistance. The function of the circulation is to service the needs of the body tissues, to transport nutrients to the body tissues, to transport waste products away, to conduct hormones from one part of the body to another, and, in general, to maintain an appropriate environment in all the tissue fluids of the body for optimal survival and function of the cells. The rate of blood flow through most tissues is controlled in response to tissue need for nutrients. The heart and circulation in turn are controlled to provide the necessary cardiac output and arterial pressure to cause the needed tissue blood flow. What are the mechanisms for controlling blood volume and blood flow, and how does this relate to all the other functions of the circulation? These are some of the topics and questions that we discuss in this section on the circulation. Physical characteristics of the circulation. Physical characteristics of the circulation. The circulation, shown in figure, is divided into the systemic circulation and the pulmonary circulation. Because the systemic circulation supplies blood flow to all the tissues of the body except the lungs, it is also called the greater circulation or peripheral circulation. Figure. Distribution of blood, in percentage of total blood, in the different parts of the circulatory system. A schematic diagram showing the circuitry of the cardiovascular system. The arrows show the direction of blood flow. Percentages represent the percent of cardiac output. Left and right sides of the heart. The right heart and the pulmonary arteries, capillaries, and veins are collectively called the pulmonary circulation. The right ventricle pumps blood to the lungs. The left heart and right heart function in series so that blood is pumped sequentially from the left heart to the systemic circulation, to the right heart, to the pulmonary circulation, and then back to the left heart. The rate at which blood is pumped from either ventricle is called the cardiac output. Because the two sides of the heart operate in series, the cardiac output of the left ventricle equals the cardiac output of the right ventricle in the steady state. The rate at which blood is returned to the atria from the veins is called the venous return. Again, because the left heart and the right heart operate in series, venous return to the left heart equals venous return to the right heart in the steady state. Finally, in the steady state, cardiac output from the heart equals venous return to the heart. Repeat and remember, the two sides of the heart operate in series, the cardiac output of the left ventricle equals the cardiac output of the right ventricle in the steady state. Remember the new term, venous return. An explanation of the circled numbers. Step 1. Oxygenated blood fills the left ventricle. Blood that has been oxygenated in the lungs returns to the left atrium via the pulmonary vein. This blood then flows from the left atrium to the left ventricle through the mitral valve, the AV valve of the left heart. Step 2. Blood is ejected from the left ventricle into the aorta. Blood leaves the left ventricle through the aortic valve, the semilunar valve of the left side of the heart, which is located between the left ventricle and the aorta. When the left ventricle contracts, the pressure in the ventricle increases, causing the aortic valve to open and blood to be ejected forcefully into the aorta. As noted previously, the volume of blood ejected from the left ventricle per unit time is called the cardiac output. Blood then flows through the arterial system, driven by the pressure created by contraction of the left ventricle. Step 3. Cardiac output is distributed among various organs. The total cardiac output of the left heart is distributed among the organ systems via sets of parallel arteries. Thus simultaneously, approximately 15% of the cardiac output is delivered to the brain via the cerebral arteries, 5% is delivered to the heart via the coronary arteries, 25% is delivered to the kidneys via the renal arteries, and so forth. Given this parallel arrangement of the organ systems, it follows that the total systemic blood flow must equal the cardiac output. The percentage distribution of cardiac output among the various organ systems is not fixed, however. For example, during strenuous exercise, the percentage of the cardiac output going to skeletal and cardiac muscle increases, compared with the percentages at rest. There are three major mechanisms for achieving such changes in blood flow to an organ system. In the first mechanism, the cardiac output remains constant, but the blood flow is redistributed among the organ systems by the selective alteration of arteriolar resistance. In this scenario, blood flow to one organ can be increased at the expense of blood flow to other organs. In the second mechanism, the cardiac output increases or decreases, but the percentage distribution of blood flow among the organ systems is kept constant. Finally, in a third mechanism, a combination of the first two mechanisms occurs in which both cardiac output and the percentage distribution of blood flow are altered. This third mechanism is used, for example, in the response to strenuous exercise. Blood flow to skeletal and cardiac muscle increases to meet the increased metabolic demand by a combination of increased cardiac output and increased percentage distribution to skeletal and cardiac muscle. Step 4. Blood flow from the organs is collected in the veins. The blood leaving the organs is venous blood and contains waste products from metabolism, such as carbon dioxide, CO2. This mixed venous blood is collected in veins of increasing size and finally in the largest vein, the vena cava. The vena cava carries blood to the right heart. Step 5. Venous return to the right atrium. Because the pressure in the vena cava is higher than in the right atrium fills with blood, called the venous return. In the steady state, venous return to the right atrium equals cardiac output from the left ventricle. Step 6. Mixed venous blood fills the right ventricle. Mixed venous blood flows from the right atrium to the right ventricle through the AV valve in the right heart, the tricuspid valve. Step 7. Blood is ejected from the right ventricle into the pulmonary artery. When the right ventricle contracts, blood is ejected through the pulmonic valve, the semilunar valve of the right side of the heart, into the pulmonary artery, which carries blood to the lungs. Note that the cardiac output ejected from the right ventricle is identical to the cardiac output that was ejected from the left ventricle. In the capillary beds of the lungs, oxygen, O2, is added to the blood from alveolar gas, and CiO2 is removed from the blood and added to the alveolar gas. Thus the blood leaving the lungs has more O2 and less CiO2 than the blood that entered the lungs. Step 8. Blood flow from the lungs is returned to the heart via the pulmonary vein. Oxygenated blood is returned to the left atrium via the pulmonary vein to begin a new cycle. Let's repeat the steps in one complete circuit through the cardiovascular system. Step 1. Oxygenated blood fills the left ventricle. Step 2. Blood is ejected from the left ventricle into the aorta. Step 3. Cardiac output is distributed among various organs. Step 4. Blood flow from the organs is collected in the veins. Step 5. Venous return to the right atrium. Step 6. Mixed venous blood fills the right ventricle. Step 7. Blood is ejected from the right ventricle into the pulmonary artery. Step 8. Blood flow from the lungs is returned to the heart via the pulmonary vein. Next question. Blood vessels. Blood vessels. The blood vessels have several functions. They serve as a closed system of passive conduits, delivering blood to and from the tissues where nutrients and wastes are exchanged. The blood vessels also participate actively in the regulation of blood flow to the organs. When resistance of the blood vessels, particularly of the arterioles, is altered, blood flow to that organ is altered. Blood vessels. There are 5 types of blood vessels. The arteries, which carry the blood away from the heart, the arterioles, the capillaries, where the exchange of water and chemicals between the blood and the tissues occurs, the venules, and the veins, which carry blood from the capillaries back towards the heart. The word vascular, meaning relating to the blood vessels, is derived from the Latin vas, meaning vessel. Some structures, such as cartilage, the epithelium, and the lens and cornea of the eye, do not contain blood vessels and are labeled avascular. Functional parts of the circulation. Before discussing the details of circulatory function, it is important to understand the role of each part of the circulation. The function of the arteries is to transport blood under high pressure to the tissues. For this reason, the arteries have strong vascular walls, and blood flows at a high velocity in the arteries. The arterioles are the last small branches of the arterial system. They act as control conduits through which blood is released into the capillaries. The arteriole has a strong muscular wall that can close the arteriole completely or can, by relaxing, dilate it several fold, thus having the capability of vastly altering blood flow in each tissue bed in response to the need of the tissue. The function of the capillaries is to exchange fluid, nutrients, electrolytes, hormones, and other substances between the blood and the interstitial fluid. To serve this role, the capillary walls are very thin and have numerous minute capillary pores permeable to water and other small molecular substances. The venules collect blood from the capillaries, and they gradually coalesce into progressively larger veins. The veins function as conduits for transport of blood from the venules back to the heart. Equally important, they serve as a major reservoir of extra blood. Because the pressure in the venous system is very low, the venous walls are thin. Even so, they are muscular enough to contract or expand and thereby act as a controllable reservoir for the extra blood, either a small or a large amount, depending on the needs of the circulation. Volumes of blood in the different parts of the circulation. Figure gives an overview of the circulation and lists the percentage of the total blood volume in major segments of the circulation. For instance, about 84% of the entire blood volume of the body is in the systemic circulation, and 16% are in heart and lungs. Of the 84% are in the systemic circulation, 64% is in the veins, 13% are in the arteries, and 7% are in the systemic arterioles and capillaries. The heart contains 7% of the blood, and the pulmonary vessels, 9%. Most surprising is the low blood volume in the capillaries. It is here, however, that the most important function of the circulation occurs, diffusion of substances back and forth between the blood and the tissues. Cross-sectional areas and velocities of blood flow. If all the systemic vessels of each type were put side by side, their approximate total cross-sectional areas for the average human being would be as follows. Cross-sectional areas and velocities of blood flow. Note particularly the much larger cross-sectional areas of the veins than of the arteries, averaging about 4 times those of the corresponding arteries. This explains the large storage of blood in the venous system in comparison with the arterial system. Because the same volume of blood must flow through each segment of the circulation each minute, the velocity of blood flow is inversely proportional to vascular cross-sectional area. Thus, under resting conditions, the velocity averages about 33 cm/sec in the aorta but only 1/1000 as rapidly in the capillaries, about 0.3 mm/section. However, because the capillaries have a typical length of only 0.3 to 1 mm, the blood remains in the capillaries for only 1 to 3 seconds. This short time is surprising because all diffusion of nutrient food substances and electrolytes that occurs through the capillary walls must do so in this exceedingly short time. Problem. Blood vessel A has a cross-sectional area of 1 square centimeter, and blood vessel B has a cross-sectional area of 10 square centimeters. If blood flow through the two vessels is the same, in which vessel is velocity of blood flow higher? Answer. Blood vessel A. Hint, velocity equals flow, per, area. Pressures in the various portions of the circulation. Because the heart pumps blood continually into the aorta, the mean pressure in the aorta is high, averaging about 100 mm of mercury. Also, because heart pumping is pulsatile, the arterial pressure alternates between a systolic pressure level of 120 mm of mercury and a diastolic pressure level of 80 mm of mercury, as shown on the left side of figure. Figure. Normal blood pressures in the different portions of the circulatory system when a person is lying in the horizontal position. Pressures in the various portions of the circulation. As the blood flows through the systemic circulation, its mean pressure falls progressively to about 0 mm of mercury by the time it reaches the termination of the venae cavae where they empty into the right atrium of the heart. The pressure in the systemic capillaries varies from as high as 35 mm of mercury near the arteriolar ends to as low as 10 mm of mercury near the venous ends, but their average functional pressure in most vascular beds is about 17 mm of mercury, a pressure low enough that little of the plasma leaks through the minute pores of the capillary walls, even though nutrients can diffuse easily through these same pores to the outlying tissue cells. Note, at the far right side of figure the respective pressures in the different parts of the pulmonary circulation. In the pulmonary arteries, the pressure is pulsatile, just as in the aorta, but the pressure level is far less, Pulmonary artery systolic pressure averages about 25 mm of mercury and diastolic pressure 8 mm of mercury, with a mean pulmonary arterial pressure of only 16 mm of mercury. Pressures in the various portions of the circulation. The mean pulmonary capillary pressure averages only 7 mm of mercury. Yet the total blood flow through the lungs each minute is the same as through the systemic circulation. The low pressures of the pulmonary system are in accord with the needs of the lungs, because all that is required is to expose the blood in the pulmonary capillaries to oxygen and other gases in the pulmonary alveoli. Basic theory of circulatory function. Although the details of circulatory function are complex, there are three basic principles that underlie all functions of the system. 1. The rate of blood flow to each tissue of the body is almost always precisely controlled in relation to the tissue need. When tissues are active, they need greatly increased supply of nutrients and therefore much more blood flow than when at rest, occasionally as much as 20 to 30 times the resting level. Yet the heart normally cannot increase its cardiac output more than 4 to 7 times greater than resting levels. Therefore, it is not possible simply to increase blood flow everywhere in the body when a particular tissue demands increased flow. Instead, the microvessels of each tissue continuously monitor tissue needs, such as the availability of oxygen and other nutrients and the accumulation of carbon dioxide and other tissue waste products, and these in turn act directly on the local blood vessels, dilating or constricting them, to control local blood flow precisely to that level required for the tissue activity. Also, nervous control of the circulation from the central nervous system provides additional help in controlling tissue blood flow. 2. The cardiac output is controlled mainly by the sum of all the local tissue flows. When blood flows through a tissue, it immediately returns by way of the veins to the heart. The heart responds automatically to this increased inflow of blood by pumping it immediately into the arteries from whence it had originally come. Thus, the heart acts as an automaton, responding to the demands of the tissues. The heart, however, often needs help in the form of special nerve signals to make it pump the required amounts of blood flow. 3. In general, the arterial pressure is controlled independently of either local blood flow control or cardiac output control. The circulatory system is provided with an extensive system for controlling the arterial blood pressure. For instance, if at any time the pressure falls significantly below the normal level of about 100 mm of mercury, within seconds a barrage of nervous reflexes elicits a series of circulatory changes to raise the pressure back toward normal. The nervous signals especially, a. increase the force of heart pumping, b. cause contraction of the large venous reservoirs to provide more blood to the heart, and c. cause generalized constriction of most of the arterioles throughout the body so that more blood accumulates in the large arteries to increase the arterial pressure. Then, over more prolonged periods, hours, and days, the kidneys play an additional major role in pressure control, both by secreting pressure-controlling hormones and by regulating the blood volume. Thus, in summary, the needs of the individual tissues are served specifically by the circulation. Thus, there are three basic principles that underlie all functions of the system. 1. The rate of blood flow to each tissue of the body is almost always precisely controlled in relation to the tissue need. 2. The cardiac output is controlled mainly by the sum of all the local tissue flows. 3. In general, the arterial pressure is controlled independently of either local blood flow control or cardiac output control. Interrelationships among pressure, flow, resistance. Blood flow through a blood vessel is determined by two factors. 1. Pressure difference of the blood between the two ends of the vessel, also sometimes called pressure gradient, along the vessel, which is the force that pushes the blood through the vessel. 2. The impediment to blood flow through the vessel, which is called vascular resistance. Figure demonstrates these relationships, showing a blood vessel segment located anywhere in the circulatory system. Figure. Interrelationships among pressure, resistance, and blood flow. P1 represents the pressure at the origin of the vessel. At the other end, the pressure is P2. Resistance occurs as a result of friction between the flowing blood and the intravascular endothelium all along the inside of the vessel. The flow through the vessel can be calculated by the following formula, which is called Ohm's law, in which F is blood flow, delta P is the pressure difference, P1 minus P2, between the two ends of the vessel, and R is the resistance. This formula states, in effect, that the blood flow is directly proportional to the pressure difference but inversely proportional to the resistance. Note that it is the difference in pressure between the two ends of the vessel, not the absolute pressure in the vessel, that determines rate of flow. For example, if the pressure at both ends of a vessel is 100 mm of mercury and yet no difference exists between the two ends, there will be no flow despite the presence of 100 mm of mercury pressure. Ohm's law, presented in the equation above, expresses the most important of all relationships that must be understood to understand the hemodynamics of the blood circulation. Let's repeat this equation. Blood flow, F, is equal to delta P divided by resistance to blood flow, R. In view of the extreme importance of this formula, the reader should become familiar with its other algebraic forms. The resistance to blood flow, R, is delta P, pressure difference of the blood between the two ends of the vessel, divided by blood flow, F. And delta P, pressure difference of the blood between the two ends of the vessel, is blood flow, F, multiplied by the resistance to blood flow, R. Relationships between blood flow, pressure, and resistance. Sample problem. Renal blood flow is measured by placing a flow meter on a woman's left renal artery. Simultaneously, pressure probes are inserted in her left renal artery and left renal vein to measure pressure. Renal blood flow measured by the flow meter is 500 milliliters per minute. The pressure probes measure renal arterial pressure as 100 milliliters per minute and renal venous pressure as 10 milliliters per minute. What is the vascular resistance of the left kidney in this woman? Solution. Blood flow to the left kidney, as measured by the flow meter, is F. The difference in pressure between the renal artery and renal vein is delta P. The resistance to flow in the renal vasculature is calculated by rearranging the blood flow equation F equals delta P divided by R, rearranging and solving for R equals delta P divided by F equals pressure in renal artery minus pressure in renal vein, divided by renal blood flow R equals 100 mm of mercury minus 10 mm of mercury, divided by 500 milliliters per min is 90 mm of mercury divided 500 mm of mercury per milliliter per minute is 0.18 mm of mercury per milliliter per minute. Problem. The renal arterial pressure is 80 mm of mercury and renal venous pressure is 10 mm of mercury. Renal blood flow of renal artery is 350 milliliters per minute. What is the vascular resistance of this kidney? Solution. Blood flow to the left kidney, as measured by the flow meter, is F. The difference in pressure between the renal artery and renal vein is delta P. The resistance to flow in the renal vasculature is calculated by rearranging the blood flow equation F equals delta P divided by R, rearranging and solving for R equals delta P divided by F equals pressure in renal artery minus pressure in renal vein, divided by renal blood flow R equals 80 mm of mercury minus 10 mm of mercury, divided by 350 milliliters per min is 70 mm of mercury divided 350 mm of mercury per milliliter per minute is 0.2 mm of mercury per milliliter per minute. Answer. The vascular resistance of the kidney is 0.2 mm of mercury per milliliter per minute. Blood flow. Blood flow means simply the quantity of blood that passes a given point in the circulation in a given period of time. Ordinarily, blood flow is expressed in milliliters per minute or liters per minute, but it can be expressed in milliliters per second or in any other unit of flow. The overall blood flow in the total circulation of an adult person at rest is about 5,000 milliliters per minute. This is called the cardiac output because it is the amount of blood pumped into the aorta by the heart each minute. Figure. A. Two fluids, one dyed red, and the other clear, before flow begins. B. The same fluids 1 second after flow begins. C. Turbulent flow, with elements of the fluid moving in a disorderly pattern. When blood flows at a steady rate through a long, smooth blood vessel, it flows in streamlines, with each layer of blood remaining the same distance from the vessel wall. Also, the central-most portion of the blood stays in the center of the vessel. This type of flow is called laminar flow or streamline flow, and it is the opposite of turbulent flow, which is blood flowing in all directions in the vessel and continually mixing within the vessel, as discussed subsequently. When laminar flow occurs, the velocity of flow in the center of the vessel is far greater than that toward the outer edges. This is demonstrated in figure. In figure A, A vessel contains two fluids, the one at the left colored by a dye and the one at the right a clear fluid, but there is no flow in the vessel. When the fluids are made to flow, a parabolic interface develops between them, as shown 1 second later in Figure B. The portion of fluid adjacent to the vessel wall has hardly moved, the portion slightly away from the wall has moved a small distance, and the portion in the center of the vessel has moved a long distance. This effect is called the parabolic profile for velocity of blood flow. The cause of the parabolic profile is the following. The fluid molecules touching the wall barely move because of adherence to the vessel wall. The next layer of molecules slips over these, the third layer over the second, the fourth layer over the third, and so forth. Therefore, the fluid in the middle of the vessel can move rapidly because many layers of slipping molecules exist between the middle of the vessel and the vessel wall. Thus, each layer toward the center flows progressively more rapidly than the outer layers. Turbulent flow of blood under some condition. When the rate of blood flow becomes too great, when it passes by an obstruction in a vessel, when it makes a sharp turn, or when it passes over a rough surface, the flow may then become turbulent, or disorderly, rather than streamline. See Figure B. Turbulent flow means that the blood flows crosswise in the vessel as well as along the vessel, usually forming whirls in the blood called eddy currents. These are similar to the whirlpools that one frequently sees in a rapidly flowing river at a point of obstruction. When eddy currents are present, the blood flows with much greater resistance than when the flow is streamlined because eddies add tremendously to the overall friction of flow in the vessel. The tendency for turbulent flow increases in direct proportion to the velocity of blood flow, the diameter of the blood vessel, and the density of the blood, and is inversely proportional to the viscosity of the blood, in accordance with the following equation, where ρ is Reynolds number and is the measure of the tendency for turbulence to occur, nu is the mean velocity of blood flow, in centimeters/second, d is the vessel diameter, in centimeters, ρ is density, and eta is the viscosity, in poise. The viscosity of blood is normally about 1/30th poise, and the density is only slightly greater than 1. When Reynolds number rises above 200 to 400, turbulent flow will occur at some branches of vessels but will die out along the smooth portions of the vessels. However, when Reynolds number rises above approximately 2000, turbulence will usually occur even in a straight, smooth vessel. Turbulent flow of blood under some conditions. Reynolds number for flow in the vascular system even normally rises to 200 to 400 in large arteries. As a result, there is almost always some turbulence of flow at the branches of these vessels. In the proximal portions of the aorta and pulmonary artery, Reynolds number can rise to several thousand during the rapid phase of ejection by the ventricles. This causes considerable turbulence in the proximal aorta and pulmonary artery where many conditions are appropriate for turbulence. 1. High velocity of blood flow. 2. Pulsatile nature of the flow. 3. Sudden change in vessel diameter. And 4. Large vessel diameter. However, in small vessels, Reynolds number is almost never high enough to cause turbulence. The transition from laminar, left, to turbulent, right, flow of water from a tap occurs as the Reynolds number increases. Development of turbulent flow in a jet. Development of turbulent flow in a jet. Vortex flow refers to localized swirling or stagnant blood flow that has separated from the central streamlines within a vessel. Such vortices, also called flow eddies, frequently occur at vascular bifurcations and distal to areas of stenosis. Unlike turbulent regions, areas of vortex flow are composed of slowly moving currents and streamlines that are not random but are often counter-current to the main flow direction. Both turbulent flow and vortex flow create problems for Mr. Angiography. A vortex street around a cylinder. This can occur around cylinders and spheres, for any fluid, cylinder size, and fluid speed provided that it has a Reynolds number between roughly 40 and 1000. The concept was introduced by George Stokes in 1851, but the Reynolds number was named by Arnold Sommerfeld in 1908 after Osborne Reynolds, 1842 to 1912, who popularized its use in 1883. Osborne Reynolds's apparatus of 1883 demonstrating the onset of turbulent flow. The apparatus is still at the University of Manchester. Diagram from Reynolds's 1883 paper showing onset of turbulent flow. Laminar flow refers to a predictable distribution of flow velocities in layers, lamina, that parallel the vessel wall. This form of flow is idealized but is nevertheless a fairly good approximation of the flow in medium and small-sized blood vessels throughout the human circulatory system. Theoretically, the distribution of velocities in a perfectly straight, non-branching vessel with non-pulsatile flow should be parabolic in cross-section, with peak velocities at the center of the lumen. In real vessels, however, the flow profile is usually more blunted because of elasticity and pulsatility effects, even though the general laminar pattern may be maintained. When the flow profile becomes flattened in this form with a nearly uniform distribution of velocities across the lumen, the term plug flow is sometimes applied. Turbulent flow is a chaotic form of fluid transport in which velocity components randomly fluctuate. Turbulence takes place when blood velocities exceed a critical threshold or when vascular morphology creates conditions that disrupt the laminar flow state. In the human circulatory system, turbulent flow is seen in the aorta, in the region of vascular bifurcations, and distal to areas of stenosis. Blood pressure. Standard units of pressure. Blood pressure almost always is measured in millimeters of mercury because the mercury manometer, shown in figure, has been used since antiquity as the standard reference for measuring pressure. Figure. Mercury barometer. Standard units of pressure. Figure. Recording arterial pressure with a mercury manometer, a method that has been used in the manner shown for recording pressure throughout the history of physiology. Standard units of pressure. Actually, blood pressure means the force exerted by the blood against any unit area of the vessel wall. When one says that the pressure in a vessel is 50 millimeters of mercury, one means that the force exerted is sufficient to push a column of mercury against gravity up to a level 50 millimeters of mercury. If the pressure is 100 millimeters of mercury, it will push the column of mercury up to 100 millimeters. Occasionally, pressure is measured in centimeters of water. A pressure of 10 centimeters of water means a pressure sufficient to raise a column of water against gravity to a height of 10 centimeters. 1 millimeter of mercury pressure equals 1.36 centimeters water pressure because the specific gravity of mercury is 13.6 times that of water, and 1 centimeter is 10 times as great as 1 millimeter. Mercury-free classic tonometer. Figure. Recording arterial pressure with a mercury manometer, a method that has been used in the manner shown for recording pressure throughout the history of physiology. High-fidelity methods for measuring blood pressure. The mercury in the mercury manometer has so much inertia that it cannot rise and fall rapidly. For this reason, the mercury manometer, although excellent for recording steady pressures, cannot respond to pressure changes that occur more rapidly than about one cycle every 2 to 3 seconds. Whenever it is desired to record rapidly changing pressures, some other type of pressure recorder is needed. Figure demonstrates the basic principles of 3 electronic pressure transducers commonly used for converting blood pressure and/or rapid changes in pressure into electrical signals and then recording the electrical signals on a high-speed electrical recorder. Each of these transducers uses a very thin, highly stretched metal membrane that forms one wall of the fluid chamber. The fluid chamber in turn is connected through a needle or catheter to the blood vessel in which the pressure is to be measured. When the pressure is high, the membrane bulges slightly, and when it is low, it returns toward its resting position. Figure. Principles of 3 types of electronic transducers for recording rapidly changing blood pressures, explained in the text. In Figure A, a simple metal plate is placed a few hundredths of a centimeter above the membrane. When the membrane bulges, the membrane comes closer to the plate, which increases the electrical capacitance between these two, and this change in capacitance can be recorded using an appropriate electronic system. In Figure B, a small iron slug rests on the membrane, and this can be displaced upward into a center space inside an electrical wire coil. Movement of the iron into the coil increases the inductance of the coil, and this, too, can be recorded electronically. Finally, in Figure C, a very thin, stretched resistance wire is connected to the membrane. When this wire is stretched greatly, its resistance increases. When it is stretched less, its resistance decreases. These changes, too, can be recorded by an electronic system. With some of these high-fidelity types of recording systems, pressure cycles up to 500 cycles per second have been recorded accurately. In common use are recorders capable of registering pressure changes that occur as rapidly as 20 to 100 cycles per second, in the manner shown on the recording paper in Figure C. Resistance to blood flow. Units of resistance. Resistance is the impediment to blood flow in a vessel, but it cannot be measured by any direct means. Instead, resistance must be calculated from measurements of blood flow and pressure difference between 2 points in the vessel. If the pressure difference between 2 points is 1 mm of mercury and the flow is 1 milliliters per second, the resistance is said to be 1 peripheral resistance unit, usually abbreviated P/R/U. Expression of resistance in C/Gs units. Occasionally, a basic physical unit called the C/Gs, centimeters, grams, seconds, unit is used to express resistance. This unit is dynesecond per centimeter to the 5th power. Resistance in these units can be calculated by the following formula. Total peripheral vascular resistance and total pulmonary vascular resistance. The rate of blood flow through the entire circulatory system is equal to the rate of blood pumping by the heart, that is, it is equal to the cardiac output. In the adult human being, this is approximately 100 milliliters per second. The pressure difference from the systemic arteries to the systemic veins is about 100 millimeters of mercury. Therefore, the resistance of the entire systemic circulation, called the total peripheral resistance, is about 100 per 100, or 1 P/R/U. In conditions in which all the blood vessels throughout the body become strongly constricted, the total peripheral resistance occasionally rises to as high as 4 P/R/U. Conversely, when the vessels become greatly dilated, the resistance can fall to as little as 0.2 P/R/U. In the pulmonary system, the mean pulmonary arterial pressure averages 16 millimeters of mercury and the mean left atrial pressure averages 2 millimeters of mercury, giving a net pressure difference of 14 milliliters. Therefore, when the cardiac output is normal at about 100 milliliters per second, the total pulmonary vascular resistance calculates to be about 0.14 P/R/U, about 1/7 that in the systemic circulation. Conductance of blood in a vessel and its relation to resistance. Conductance is a measure of the blood flow through a vessel for a given pressure difference. This is generally expressed in terms of milliliters per second per millimeter of mercury pressure, but it can also be expressed in terms of liters per second per millimeter of mercury or in any other units of blood flow and pressure. It is evident that conductance is the exact reciprocal of resistance in accord with the following equation. Conductance equals 1 per resistance. Very slight changes in diameter of a vessel can change its conductance tremendously. Slight changes in the diameter of a vessel cause tremendous changes in the vessel's ability to conduct blood when the blood flow is streamlined. This is demonstrated by the experiment illustrated in Figure, which shows 3 vessels with relative diameters of 1, 2, and 4 but with the same pressure difference of 100 millimeters of mercury between the 2 ends of the vessels. Although the diameters of these vessels' conductance resistance increase only 4-fold, the respective flows are 1, 16, and 256 milliliters per minute, which is a 256-fold increase in flow. Thus, the conductance of the vessel increases in proportion to the 4th power of the diameter, in accordance with the following formula. Conductance proportionally diameter to the 4th power. Figure A. Demonstration of the effect of vessel diameter on blood flow. B. Concentric rings of blood flowing at different velocities. The farther away from the vessel wall, the faster the flow. Poiseuille's law. The cause of this great increase in conductance when the diameter increases can be explained by referring to Figure, which shows cross-sections of a large and a small vessel. The concentric rings inside the vessels indicate that the velocity of flow in each ring is different from that in the adjacent rings because of laminar flow, which was discussed earlier in the lecture. That is, the blood in the ring touching the wall of the vessel is barely flowing because of its adherence to the vascular endothelium. The next ring of blood toward the center of the vessel slips past the first ring and, therefore, flows more rapidly. The third, fourth, fifth, and sixth rings likewise flow at progressively increasing velocities. Jean-Leonard Marie Poiseuille, 1797–1869, was a French physicist and physiologist. Thus, the blood that is near the wall of the vessel flows extremely slowly, whereas that in the middle of the vessel flows extremely rapidly. In the small vessel, essentially all the blood is near the wall, so that the extremely rapidly flowing central stream of blood simply does not exist. By integrating the velocities of all the concentric rings of flowing blood and multiplying them by the areas of the rings, one can derive the following formula, known as Poiseuille's law, in which F is the rate of blood flow, delta P is the pressure difference between the ends of the vessel, R is the radius of the vessel, L is length of the vessel, and eta is viscosity of the blood. Note particularly in this equation that the rate of blood flow is directly proportional to the fourth power of the radius of the vessel, which demonstrates once again that the diameter of a blood vessel, which is equal to twice the radius, plays by far the greatest role of all factors in determining the rate of blood flow through a vessel. Importance of the vessel diameter, fourth power law, in determining arteriolar resistance. In the systemic circulation, about 2/3 of the total systemic resistance to blood flow is arteriolar resistance in the small arterioles. The internal diameters of the arterioles range from as little as 4 micrometers to as great as 25 micrometers. However, their strong vascular walls allow the internal diameters to change tremendously, often as much as fourfold. From the fourth power law discussed above that relates blood flow to diameter of the vessel, one can see that a fourfold increase in vessel diameter can increase the flow as much as 256-fold. Thus, this fourth power law makes it possible for the arterioles, responding with only small changes in diameter to nervous signals or local tissue chemical signals, either to turn off almost completely the blood flow to the tissue or at the other extreme to cause a vast increase in flow. Indeed, ranges of blood flow of more than 100-fold in separate tissue areas have been recorded between the limits of maximum arteriolar constriction and maximum arteriolar dilatation. Blood pumped by the heart flows from the high-pressure part of the systemic circulation, i.e., aorta, to the low-pressure side, i.e., vena cava, through many miles of blood vessels arranged in series and in parallel. The arteries, arterioles, capillaries, venules, and veins are collectively arranged in series. When blood vessels are arranged in series, flow through each blood vessel is the same and the total resistance to blood flow, Rtotal, is equal to the sum of the resistances of each vessel. Rtotal equals R1 plus R2 plus R3 plus R4. The total peripheral vascular resistance is therefore equal to the sum of resistances of the arteries, arterioles, capillaries, venules, and veins. In the example shown in figure, the total vascular resistance is equal to the sum of R1 and R2. Resistance to blood flow in series and parallel vascular circuits. Blood vessels branch extensively to form parallel circuits that supply blood to the many organs and tissues of the body. This parallel arrangement permits each tissue to regulate its own blood flow, to a great extent, independently of flow to other tissues. For blood vessels arranged in parallel figure, the total resistance to blood flow is expressed as 1 per R total equals 1 per R 1 plus 1 per R 2 plus 1 per R 3 plus 1 per R 4. Problem. 3 vessel resistors, each with a value of 10, are arranged in parallel. How much does total resistance change if a fourth vessel resistor with a value of 10 is added in parallel? Possible answers. A. Total resistance decreases from 3.33 to 2.5. B. Total resistance increases from 30 to 40. C. Total resistance decreases from 30 to 20. D. Total resistance does not change. Answer. Total resistance decreases from 3.33 to 2.5. It is obvious that for a given pressure gradient, far greater amounts of blood will flow through this parallel system than through any of the individual blood vessels. Therefore, the total resistance is far less than the resistance of any single blood vessel. Flow through each of the parallel vessels in figure is determined by the pressure gradient and its own resistance, not the resistance of the other parallel blood vessels. However, increasing the resistance of any of the blood vessels increases the total vascular resistance. It may seem paradoxical that adding more blood vessels to a circuit reduces the total vascular resistance. Many parallel blood vessels, however, make it easier for blood to flow through the circuit because each parallel vessel provides another pathway, or conductance, for blood flow. The total conductance C, total, winky face, for blood flow is the sum of the conductance of each parallel pathway. C, total, equals C1 plus C2 plus C3 plus C4. For example, brain, kidney, muscle, gastrointestinal, skin, and coronary circulations are arranged in parallel, and each tissue contributes to the overall conductance of the systemic circulation. Blood flow through each tissue is a fraction of the total blood flow, cardiac output, and is determined by the resistance, the reciprocal of conductance, for blood flow in the tissue, as well as the pressure gradient. Therefore, amputation of a limb or surgical removal of a kidney also removes a parallel circuit and reduces the total vascular conductance and total blood flow, i.e., cardiac output, while increasing total peripheral vascular resistance. Effect of blood hematocrit and blood viscosity on vascular resistance and blood flow. Note especially that another of the important factors in Poiseuille's equation is the viscosity of the blood. The greater the viscosity, the less the flow in a vessel if all other factors are constant. Furthermore, the viscosity of normal blood is about 3 times as great as the viscosity of water. But what makes the blood so viscous? It is mainly the large numbers of suspended red cells in the blood, each of which exerts frictional drag against adjacent cells and against the wall of the blood vessel. Hematocrit. The percentage of the blood that is cells is called the hematocrit. Thus, if a person has a hematocrit of 40, this means that 40% of the blood volume is cells and the remainder is plasma. The hematocrit of men averages about 42, while that of women averages about 38. These values vary tremendously, depending on whether the person has anemia, on the degree of bodily activity, and on the altitude at which the person resides. Hematocrit is determined by centrifuging blood in a calibrated tube, as shown in figure. The calibration allows direct reading of the percentage of cells. Figure. Hematocrits in a healthy, normal, person and in patients with anemia and polycythemia. Effect of blood hematocrit and blood viscosity on vascular resistance and blood flow. Effect of hematocrit on blood viscosity. The viscosity of blood increases drastically as the hematocrit increases, as shown in figure. The viscosity of whole blood at normal hematocrit is about 4. This means that 4 times as much pressure is required to force whole blood as to force water through the same blood vessel. Figure. Effect of hematocrit on blood viscosity. Water viscosity equals 1. When the hematocrit rises to 60 or 70, which it often does in polycythemia, the blood viscosity can become as great as 10 times that of water, and its flow through blood vessels is greatly retarded. Problem. What are the hematocrit and viscosity for patients A, B, and C? See picture. Solvation. Patient A, hematocrit equals 30, blood viscosity equals 3. Patient B, hematocrit equals 50, blood viscosity equals 5. Patient C, hematocrit equals 60, blood viscosity equals 7. See the figure. Problem. What are the hematocrit and viscosity of patient A, B and C? See the figures. Solvation. Patient A, hematocrit equals 50, blood viscosity equals 5. Patient B, hematocrit equals 30, blood viscosity equals 3. Patient C, hematocrit equals 60, blood viscosity equals 7. See the figure. Other factors that affect blood viscosity are the plasma protein concentration and types of proteins in the plasma, but these effects are so much less than the effect of hematocrit that they are not significant considerations in most hemodynamic studies. The viscosity of blood plasma is about 1.5 times that of water. Effects of pressure on vascular resistance and tissue blood flow. From the discussion thus far, one might expect an increase in arterial pressure to cause a proportionate increase in blood flow through the various tissues of the body. However, the effect of pressure on blood flow is greater than one would expect, as shown by the upward curving lines in figure. Figure. Effect of arterial pressure on blood flow through a blood vessel at different degrees of vascular tone caused by increased or decreased sympathetic stimulation of the vessel. The reason for this is that an increase in arterial pressure not only increases the force that pushes blood through the vessels but also distends the vessels at the same time, which decreases vascular resistance. Thus, greater pressure increases the flow in both of these ways. Therefore, for most tissues, Blood flow at 100 mm of mercury. Arterial pressure is usually 4 to 6 times as great as blood flow at 50 mm of mercury instead of 2 times, as would be true if there were no effect of increasing pressure to increase vascular diameter. Note also in figure the large changes in blood flow that can be caused by either increased or decreased sympathetic nerve stimulation of the peripheral blood vessels. Thus, as shown in the figure, inhibition of sympathetic activity greatly dilates the vessels and can increase the blood flow twofold or more. Conversely, very strong sympathetic stimulation can constrict the vessels so much that blood flow occasionally decreases to as low as 0 for a few seconds despite high arterial pressure. Problem. Where am I? For each item in the following list, give its correct location in the graph. Normal. Sympathetic stimulation. Sympathetic inhibition. Parasympathetic stimulation. Parasympathetic inhibition. Solvation. Normal is B. Sympathetic stimulation is C. Sympathetic inhibition is A. Parasympathetic stimulation is not represented on the graph. Parasympathetic inhibition is not represented on the graph. Thank you for attention.